Welcome. This is lecture number 2, and in this lecture it's entitled The Microscope, the Human Bloodstream and Cystic Borrelia Attack Models, Shape-Shifting Form Metamorphosis, Non-Spiral Borrelia, and it is a continuation of what we have covered in lecture 1, which is an introduction to the cystic forms of Borrelia, which are non-spiral, but which are real and which are not covered adequately in the textbooks. The lecture in this portion will focus on the microscope and its use in detecting parasites of Borrelia in the human bloodstream, cystic Borrelia, the attack models of cystic Borrelia as they interact with human tissue and other mammalian tissues, and an overview of what really counts in the living body when you're dealing with Borrelia infection. It's not always that you're dealing with spiral forms under the microscope. You may be dealing with cystic and other forms. Those other forms also include granular forms, cell-wall deficient forms. They will be covered in future lectures, but this lecture will deal with more on the cystic Borrelia. So as an overview, we will extend the lessons learned from cystic Borrelia and carry these forward to dissemination. Remember that Borrelia infections are acquired in a blood-to-blood scenario, and that means that the infection is first carried by the blood of a small mammal or a large mammal. The tick takes a blood meal. For a time, the spirochetes then live inside the tick gut, and then if fate has it, the tick will feed on the human and transmit the infection, the spirochetes or other forms of Borrelia, into the human bloodstream. So it goes from the blood of the small mammal, which is a reservoir, or the deer, which is a reservoir, to the human mammalian host. And during this time, cystic forms may appear, and we'll consider how they look under the microscope and what the variables are in terms of recognizing correctly cystic Borrelia. Cystic Borrelia are not discussed in any major textbook, and they are more or less ignored by the prevailing publishers of microbiology and medical textbooks today. So we're going to try to close that deficiency and make up for the oversights and give you a better idea of what is happening with the big picture of Borrelia, which includes cystic forms. Now, when you're traveling by air and you're approaching your destination airport, the aircraft flight attendant will also be careful to warn you that when you open the overhead compartment, you should be careful because some of the objects which you place in the overhead compartment may have shifted. They may have changed place, and they may fall on your head or injure you or the person next to you as you open the overhead compartment. So what they say is shift happens. And I'm going to follow up that motif with this cartoon, and that is that shift happens in nature. And in this situation, with the model of butterfly, the pupa, which is shown here, undergoes a metamorphosis and becomes a butterfly. This is part of the developmental normal cycle of the life of the butterfly pupa unit. They don't look anything like each other, but they are related to each other genetically, and one follows the other as A follows B, or one follows two. They are linked, they are a logical unit, and you have to be able to identify both the pupa form, or the caterpillar form, and the butterfly form. They're the same organism, but they look different. Now, as part of Lecture 1 content, we discussed the spiral form of Borrelia and how under adverse conditions, the spiral form can transform into rounded cystic structures or round bodies. And we also discussed that these round bodies, under proper nutrient conditions, can reverse the process and reform the spiral forms of the Borrelia, which is the form that is best described in textbooks and which we are most familiar with. The spiral form is real. The cystic form is real in any of its sizes. They are interconvertible, and they contain the same DNA. The cystic form can cause disease and can persist in reservoirs in the host during the dormant period when no infection is being transmitted, no blood meals are being taken. The other form, the cystic form, can also appear in the reservoir hosts. It can cause disease. It can persist in the host during times of starvation or adverse biological environments. And this rounded up cystic form in any of its sizes is a survival adaptation of the spiral form to adverse conditions. When the conditions are good, then the reconversion occurs and the spiral form reforms from the cystic form. So there is complete reversibility, complete equality. This can cause disease, the spiral form, and we'll see that the cystic form too can cause disease in the human host. Our first order of business is a critical review of blood studies, and blood studies are the alpha and the omega, the beginning and the end of many people's experience with diagnosis in Lyme disease. The blood studies are not perfect, and blood studies have diagnostic shortcomings from the existing blood examination methods. People will ask, after they've been to the doctor and a blood test was obtained to see if they have Lyme disease or borreliosis, what did the blood test show? And that's what's going on today in 2013. What did the blood test show? The expectation is that if the blood test says yes, we detected sufficient evidence for antibodies to Lyme disease, then you have the problem, you have the infection, you're getting antibiotics, you undergo treatment, you'll get better. If the blood test turns up a negative result, then you'll be told that you don't have the infection, you won't get antibiotics, you're on your own, and whatever symptoms you have are not related to Lyme disease, and there's nothing more to do in terms of what the doctor can offer you, other than perhaps reassure you, recommend that you seek other specialties like psychiatry or pain management, and certainly you won't get antibiotics if you don't present with a blood test result that's positive. Now, one exception to that rule is that if the doctor and the patient both witness the characteristic erythema migrans rash, which is what we all hear about, that's the rash that occurs at the site of the tick bite, and it's said to be a bullseye type rash. That's not true. The erythema migrans rash has at least 17 to 18 different varieties, and the bullseye type is in the minority of all the other 18 varieties. It can be a red patch that's all red with no target. It can be very pale. It can be ringworm-like with a red edge. It can look vesicular and look like poison ivy. It can be resembling a bruise. It can be purpuric. It can have crust and central necrosis. It can have appearance which is very temporary and fleeting. We call it transient or evanescent erythema migrans. You'll see it in the morning. You'll report to your doctor's office, and you'll unroll the sleeve or lift up your pant leg, and the rash will be gone. And the doctor will not be able to confirm that what you saw in the morning is actually helpful in diagnosing Lyme disease. So in this situation, you'll either get a blood test or be referred for a follow-up appointment a period 6 weeks later when blood will be drawn, and they'll see what the blood test showed. And then the goal is to hopefully find antibodies in sufficient level of the right kind to diagnose whether or not you have Lyme disease. If you don't have the antibodies in the blood test, then the current thinking is that you do not have the problem, and that whatever is bothering you is not Lyme disease. That may be reasonable in some situations, but it may be very unreasonable in other situations where Lyme disease exists, but your blood test shows a negative result. So here are your menu choices with blood tests. Menu choices are, do you have antibodies? And in order to measure those, the ELISA test will be done first. And if the ELISA test has a number that's high enough, you may or may not be referred to a second test, which is called the Western blot test for bands. And if those bands are of the correct type and the correct number according to CDC criteria, usually you like to see 5 bands. In some situations less, but mostly they're looking for 5 bands on the Western blot. Then you will be reassured that your correct diagnosis is that you have Lyme antibodies in your blood, and you'll get antibiotics, and hopefully those antibiotics will be given in a dose and a time over a time period that will give you relief and give you a chance for cure. Now, if the antibody test is negative and it's in a borderline, maybe, I don't know, but it's there somewhat, it's not high enough to call, then borderline patients will be referred sometimes by specialists to get a DNA test for the detection of Borrelia DNA. And this test is called a Pcr test, or polymerase chain reaction. And the goal of this test is to amplify Borrelia DNA. And of course, the DNA of the Borrelia is very complicated, as we showed in the first lecture. And so a PCR test is only able to try to amplify portions of the DNA of the Borrelia. The main chromosome has about 1,000 different bases, and there are 22 chromosomes and many chromosomes, and those are called plasmids. They each have DNA, and so the PCR test may be designed to amplify either the chromosome type DNA or the plasmid DNA. It is said that if a properly performed PCR test is negative, and remember, only portions of the DNA, either chromosomal or plasmid, can be evaluated by any PCR test. But if your PCR test, whether it's chromosomal or plasmid, is negative, then they'll say, well, there's great doubt that you have Lyme disease, your antibodies are so-so, your PCR didn't amplify, we don't have proof that you have Borrelia DNA, therefore you must have some other condition, and you will not get antibiotics, and you will not be diagnosed with Borrelia or Lyme infection. So we have now 2 of the menu choices covered. We have the antibody part, which is ELISA, and Western blot. And now we have the second, more technical type of testing, which is the PCR test, looking for evidence of DNA or RNA of the Borrelia, and hoping to amplify that, but they can only amplify portions of it. And any one PCR test will not amplify all of the possible portions. And we learned in the last lecture that the DNA of Borrelia is a DNA in flux and changing. So your PCR test may not, with primers, which start the test going, amplify successfully the Borrelia that's in your body. And you'll turn up with a negative test, even though you have a Borrelia that doesn't get amplified by the DNA PCR test. So a third pathway to diagnosis is the old-fashioned pathway, and that is to look for actual Borrelia microbes in the blood under the microscope, looking for organisms in the blood. And you can do that by doing blood cultures, and blood cultures now are coming into their own. We use cultures of blood for diagnosis of many microbiological conditions. Many infectious diseases depend on the results of blood cultures. We look for the actual microbes in the blood. They grow in the test tube, they grow in artificial media, and then we can detect them in the microbiology laboratory. Another way of looking for microbes in the blood or parasites in the blood is to look directly into the microscope with a blood smear and to see if you can see those microbes with the microscope in a blood smear. This is more tedious. It has not come into general use with Lyme disease, but there is progress being made in some laboratories around the world, which may help to make the microscopic examination of blood smears for Borrelia microbes a practical tool, not now, but in the future. And I'm going to show you some of those techniques and some of those results in this lecture. So, blood tests, great if they're positive. If they're not positive, it's not absolutely conclusive that you're a negative Lyme patient. Your antibodies may not match the test strip. Your DNA may not match the PCR primers. And so these two technologies may miss it. Blood cultures may find it. And microscope examination may also find it in the blood, as I will show you in the following slides. So we are going to be looking at blood. And our field of view, that's what we call the area under the microscope where we can see anything at all, field of view. It's a round area the microscope allows to be in focus at any one time. It's very, very narrow. And in the conventional field of view, which is dependent on detection of antibodies, the antibody detection is very, very narrow too. Because as I told you, the B31 strain, which is used for all of USA blood testing for antibodies, is a strain which does not necessarily match the 100 other strains, genotypes of Borrelia, which are out there. So the field of view is negative for the microscope, and it's very, very narrow and limited for detection of antibodies. In the 30 years of time since the discovery of Lyme disease spirochete, antibodies have ruled. They have centered all of the attention by doctors and by researchers on the diagnosis of Borrelia burgdorferi diseases. Do you have an antibody? If you don't have an antibody, the conventional teaching is you don't have the disease. In medical terms, if you don't have the antibody, you are sero, that's antibody, serology, negative. So seronegative. Now, does seronegative mean that you don't have Lyme disease? No, it does not, because there are many examples published in the literature where people have Lyme disease or related Borrelia infections where there is no antibody detected in the blood, but they do have the disease, and they can find it with other methods. They can biopsy it, they can culture it, and there are other ways for diagnosing it. You can find the erythema migrans rash and see that witnessed by a physician. So antibodies are not the be-all and end-all for the diagnosis of blood. Blood, antibody, one choice. But allowing antibodies to be the be-all and end-all for diagnosis is lazy. It's a lazy approach to Borrelia medicine. The idea that no antibodies equals no disease, I put sick around here, that means that I don't think that that sentence is correct. No antibodies does not prevent you from having Lyme disease or a related Borrelia infection. And the greatest simple example of that is the recent discovery of Borrelia miyamotoi, which has now been reported in the New England Journal of Medicine. It's a Borrelia infection, somewhat related to Borrelia burgdorferi and Lyme disease, but it doesn't react in any of the blood tests available today in the United States. So you can have a spirochete, Borrelia miyamotoi, and the blood test will show no antibodies, but you have real disease. Not no disease, real disease. Your antibody test, your blood test, negative. There is no blood test right now for Miyamoto Borrelia. So, medicine is a healing art. And we become physicians and we learn the healing art in medical school and residency and further training by learning from our predecessors. And learning medicine is much more than reading the textbook and memorizing what's in the medical textbook. You have to learn how to apply the art or the healing art of medicine to patient care. It's far more complicated. And it's much more complicated with machines reading blood antibodies under a laboratory situation. Medicine is a healing art. Medicine relies on patient examination, taking a history, doing a physical examination, thinking about the patient's situation, listening to what the patient tells you, making sense of what they tell you, and then reconciling all that with a list of possible diagnoses. We call it differential diagnoses. And then one by one, either confirming or excluding any of those diagnoses. Microscopic direct examination of the blood is an old-fashioned, state-of-the-art tool from parasitology. So if you had malaria as one of your differential diagnoses, direct examination of the blood would be a preferred method of diagnosing malaria because the parasites appear, they appear in the blood, and they're most easily identified when the patient has spikes of fever. You draw the blood during the fever spikes, you look for the malaria parasites, and if you find the parasites, then you can make a diagnosis of malaria. There is now underway a team of researchers who want to apply the direct blood examination, which is used in malaria and which is used in the diagnosis of other parasitic diseases such as sleeping sickness and Chagas disease and other parasitic diseases that involve blood, leishmaniasis, to Borrelia infections and to the diagnosis of other co-infections, which are not Borrelia, but infections which are transmitted by the same tick which transmits the Borrelia. They travel with the Borrelia, and these co-infections include babesiosis, anaplasmosis, ehrlichiosis, and other parasitic infections that can be transmitted at the same time as the Borrelia is transmitted. So one tick bite can give you 2, 3, 4, 5 infections simultaneously. So discovery infections in the blood by diagnosis, by microscopic examination, seeing the parasites in the blood, in some situations gives you a diagnosis. It is very helpful in the co-infections. When you see the proper changes in the blood, you can make a diagnosis of babesiosis, or you can make a diagnosis of ehrlichiosis, or bartonellosis, or anaplasmosis, or other co-infections which travel with Lyme disease. So let's start looking at the patient blood under the microscope for God's sakes. Even if antibodies in blood are never produced. You may be gratified and the patient may be helped by a microscopic examination of the patient's blood. Now, Dr. Morten Laane is a professor at the University of Oslo in Norway, and this is Dr. Laane. You can see he's a very robust man. He uses high technology. Here's Dr. Laane at his desk looking at a computer screen, looking for parasites in the blood. And you probably guess that Dr. Laane is maybe 50, maybe 60 years old. I'm 65. He looks more robust than I do. Dr. Laane is in his 80s. And he is a fellow of the Royal Academy of Microscopy in London. This is an honorary position which very few people in their lifetime ever achieved. This tells you that the University of Oslo is very fortunate to have Dr. Laane as a fellow of the Royal Academy of Microscopy, London, England, 50 years of advanced microscopy techniques under his belt, and with a laboratory equipped with the highest resolution light microscopes in the world. Dr. Laane is a gem. Dr. Laane is a team player, and Dr. Laane is a leader and an independent thinker, very robust, and still very active at the age of 80 in his laboratory in Oslo, Norway. Now, Dr. Laane has developed a direct blood microscopic examination of Norwegian patients who have chronic, I'm saying that's chronic, borreliosis. And lots of his patients are hunters and spend their time outside in the forests of Norway and are exposed to ticks and get many tick bites. And they will, in certain cases, develop Borrelia infections of the European type. And they may develop co-infections like babesiosis or marginalosis or ehrlichiosis or anaplasmosis. And so the patients may have multiple parasites in the blood. Dr. Laane is interested in looking at their blood and seeing if he can make a diagnosis to help with the patient diagnosis and management. What has Dr. Laane found in some of the patients who have chronic borreliosis from Oslo, Norway? Well, here he is in his office, and here are the fruits of his labor. These are wonderful-looking Borrelia Spirochetes that Dr. Laane has identified by direct examination of human blood under the microscope. Now, this is unheard of, but Dr. Laane is not your usual microscopic guy. He is a fellow of the Royal Academy. He has 50 years of experience. He has the best equipment. He has a very keen mind. He's a very innovative guy. He spends a lot of time looking, and he makes lots of discoveries that are unique and are very important to our understanding and our development of Borrelia diagnosis in year 2013 and beyond. He is an extraordinary microscopy professor, and he has made new discoveries in human borreliosis infection by direct microscopic examination of human blood. Oh my God. Looking at direct human blood under the microscope, looking for Borrelia and co-infections. This is unheard of in the United States, frowned upon, scoffed at, and vilified. Now, Dr. Laane has these beautiful images to show you what the fruits of his labor are all about. He succeeds in some patients in identifying this spirochete, which is a parasite, in the blood of patients who have chronic borreliosis in Oslo, Norway. Here's one of the structures that Dr. Laane has identified in human blood. What is it? It's right here. What are we looking at? We're looking at some sort of a snake-like structure. It's inside a round structure. And I'll give you a hint. This round structure is a red blood cell from human blood. And this snake-like structure is a Borrelia spirochete. So, what has Dr. Laane identified? He's identified the Borrelia spirochete in human blood, but it has penetrated and infiltrated and set up housekeeping inside a red blood cell. So the clues, well, it's rolled up. It's inside a red blood cell. It's snake-like. It came from microscopic examination, patients from Norway, and the patient has chronic Borrelia infection. It's a Borrelia parasite inside of human blood. Unheard of in the United States, perhaps because we don't have people of the caliber of Dr. Laane looking at blood the way he looks at it with his equipment and his training experience in the United States today. Now, for comparison, this is a cystic Borrelia with a parasite inside a cyst form. And we've seen cyst forms in lecture number 1. It's a rounded body. The spirochete rolls up like the carpenter's ruler rolls up and forms a round structure. And these are all rounded up spirochetes inside a spirochetal cyst form. This is a red blood cell. This is a spirochetal cyst form. The Borrelia are the same. Now, some more of Dr. Laane's magnificent Borrelia discoveries. This is a panel of images, and it shows what some of Dr. Laane's fruits of labor, his discoveries have included from human patients in Norway with his direct blood microscopic examination. This is a cystic form of Borrelia. It has a little dot inside. It has a very distinct cell wall. There's no red blood cells around it, and I'll tell you why later on. Another cystic form of Borrelia. Another cystic form where the spirochete is becoming more elongated. Here's a cystic form where it actually looks like a hook. Here it's now looking like a snake inside a cyst. And then here is a cyst which has ruptured and the spirochete Borrelia has popped out and is going into the blood itself. Here's another one where it's ruptured. The spirochete is broken out of the cyst. Remember the wrinkled cyst that used to be bigger, now wrinkled because it's smaller. It's ruptured. The spirochete has escaped from it. And another cyst with a wrinkled wall and a spirochete hanging out into the red blood. Here's the spirochete that's completely free of the cyst. And then these are straightforward Borrelia spirochetes, all from human blood, all from direct examination. So some of the human spiral blood Borrelia are contained within cystic forms. Contained within cystic forms and Borrelia, that's old hat. We knew that in the last lecture. Cysts circulating in human blood containing Borrelia, that's new stuff. We didn't cover that in the last lecture, and that is new stuff that is brought to us from Dr. Laane. It's possible to identify such forms if you spend the time with the microscope and use his tools and his techniques. You'll be able to find cystic forms in the blood containing Borrelia, and you'll be able to see spiral forms, or cysts that are broken open. So here they are. Spiral Borrelia within cystic forms. Here's the Borrelia. Here's the cyst wall. Remember the wrinkles? Used to be bigger, now smaller. Here's a spiral form that has broken out of its cyst wall partially, and a spirochete Borrelia type that's in the shape of a hook. It's associated with a cyst from blood, and another one which is a dot-like form of Borrelia inside a cyst. It's wrinkled. Remember the wrinkles? It means the cyst used to be larger. Now it has lost some of its water and it has shrunk in volume. Wrinkled cyst, Borrelia inside. Dr. Laane, direct examination of human blood, diagnosis of Borrelia infection in humans by direct microscopic examination. No antibodies needed, no other methods needed. You have the diagnosis here under the microscope. This is incredible. So Dr. Laane has identified the breaking open part of cystic Borrelia and the release of spiral forms into the blood in his patients from Oslo, Norway. And here we are again, spirochete Borrelia breaking out of the cyst, going into the blood in a free form. Wrinkled cyst again, wrinkled cyst, Borrelia breaking out, going into the blood, and another cyst that has broken open. Let's look at the wrinkles. We saw that in lecture number 1. Spirochete has now escaped and is going into the human blood. It's broken out. Now, Dr. Laane in Norwegian uses a word which is translated as holster to describe the cyst. In our USA English, we would think of holster in a different context, but Dr. Laane thinks of the wrapper or the cyst wall, which covers the spirochete, which is breaking open, as a holster. It describes the wall of the cystic Borrelia. Now, in our language, we use holster to describe the leather that holds the gun, or the leather which holds the knife, both of which are weapons. And when the weapon is needed, then the weapon is taken out of the holster and used to assault. So, in like manner, if we look at the holster as the cyst and the spirochete breaking out of the cyst, the spirochete is getting out of the cyst and is ready to assault the human body. I don't think we need any further commentary on holster, its implications for human health. This is a weapon. This is a holster. This is here to do damage when it gets into a mammalian body. I'm going to introduce now a term called the string of pearls. This is another structure which Dr. Laane has identified under the microscope in certain human blood examinations. It's a special form of Borrelia spirochetes, not written about in any of the textbooks, and so it's a special form which is specific enough that when we see it, this is a string of pearls. Think of a pearl necklace, and each of these is one of the pearls in the necklace. Think of this as a string of pearls attached to a tail structure coming out of a cystic Borrelia. The string of pearls is a marker which tells us that we are in the presence of Borrelia. Even though we don't see cysts, even though we don't see the cyst here, we see the pearls here. They've broken away from the cyst, they're in the blood, they're free. This string of pearls is helpful. It's diagnostic and helpful in diagnosing Borrelia infection when you see it in the blood or in tissue. The Laane string of pearls type organism is found in human blood. This is a first. This has never been done before by anybody on the planet. And this is a string of pearls from one of Dr. Laane's patients in human blood. This is probably a cystic form. And so you see that the pearls have some relationship in form and shape, size, to a string of pearls. That would be a jewelry item that you would give as a gift, very expensive. But each of these pearls is connected by a string, and the pearls can break loose from the string and become free, and you have single pearls, but when they're on the string, they are a string of pearls. Now, who believes in the string of pearls concept? Well, Dr. Willy Bergdorfer, for one. He discovered the Lyme disease organism. Dr. Oystein-Rosen, and Dr. Laane, and for what it's worth, Dr. Alan MacDonald, too. I believe that the string of pearls is very important, and when you see it in tissue, when you see it in blood, it tells you that you are in the presence of a Borrelia infection. The string of pearls comes as an add-on to the tail structure, which comes out of some of the cystic forms of Borrelia. And here you see one string of pearls. Here you see a tail structure. And at the other end, you see a string of pearls, and it's coming out of the cystic structure of Borrelia. Here again, a cystic Borrelia with a coiled up spirochete, a snake-like thing coiled up inside the cyst, and the string of pearls coming out at the end of the cyst. It's often attached then to cystic Borrelia. It's associated with the tail. The tail is exposed at the edge of the cystic form. And if you look in the PubMed National Library of Medicine search engine using software Borrelia and string of pearls, references will pop up, and you'll see these in the next slide. You'll see who's writing about string of pearls and Borrelia. These are not lightweights, these are heavyweights who are writing about a string of pearls. Dr. Willy Bergdorfer. Fred Hayes, who's a senior technician, worked with Dr. Bergdorfer for 30, 40 years at the Rocky Mountain Lab, one of the experts in the ultrastructure electron microscopy of Borrelia burgdorferi. String of pearls, structured in copious amounts. Dr. Brosen and his cousin, Sverre Henning Brosen. String of pearls, right? Dr. Per Mercer, string of pearls. Dr. Bergdorfer again, and Dr. Fred Hayes. Spaghetti-like or string of pearls-like spherical forms under the electron microscope. And then, lastly, in 2004, Dr. Ali Tallo describing Borrelia burgdorferi spirochetes, and the structure resembles a string of pearls. See figures. So, all of these people, important investigators, none more important than Dr. Bergdorfer and Fred Hayes and Drs. Rosen. Nobody more important in the world. And these are the pearls, and this is the tail structure of the Borrelia. They do look like a string of pearls. Now, what's in the spirochete? DNA and proteins. What's in the pearls? DNA and proteins. Is there variability of DNA along the axis of the Borrelia? Yes, because there's more than 10 to 40 copies of the chromosome and all the plasmids as you move along the chromosome. So when it segments and makes little pearls, each one potentially has one copy complete of the chromosome and the plasmids, and each of the pearls can have a different DNA copy. So each of the pearls can have a separate DNA and protein makeup. This is a 3D view of cystic Borrelia from the work of Dr. Alban, Dr. Nelson at the University of Rhode Island. And what are we looking at? in the yellow boxes, string of pearls, string of pearls, string of pearls, coming off of the tail structure coming out of the cyst. String of pearls, here's the tail structure. And this is the way they start out. Now, they don't stay attached to the tail structure forever. They can break off. And when they break off and they become free, then you'll see the string of pearls not connected to the tail structure, but floating free in the blood. And here we have it. String of pearls, string of pearls, string of pearls attached. So what Dr. Laane has done is an amazing discovery. He's found string of pearls, Borrelia structures in human blood using his microscope and his advanced technique. String of pearls Borrelia. These are, like the other forms that I talked about, perfect Borrelia forms. No other life form on the planet produces a string of pearls in human blood. Period. This is a string of pearls. It's broken away from its tail structure. It's circulating in human blood, and Dr. Laane took a picture of it. This patient has Borrelia infection. Even though you don't see a spiral corkscrew, the string of pearls is just as diagnostic. This is definitive. Now, we have the Borrelia type string of pearls here. We have the jewelry type with the pearls that are recovered from diving by pearl divers for oysters that contain pearls. And just as an aside, we have the jazz version of the string of pearls, which is a hit from the 1940s, 1942. And I think, and I can't prove this, but I think that as a jazz enthusiast and familiar with the Glenn Miller Band and its music, I was aware of their song, String of Pearls, their record. And when I first saw these structures in electron micrographs in the Rocky Mountain lab produced by Dr. Bergdorf and Fred Hayes, I suggested the term string of pearls. Now, Dr. Bergdorf is a lot older than I am, so he may have also thought of the idea himself, but I'd like to think that I had something to do with the naming of String of Pearls after the jazz record by the Glenn Miller Band in 1942. It does look like a string of pearls. Borrelia type, not jewelry type, but musical type. Now, the string of pearls develops from the tail region of the Borrelia. It may break off from the tail and circulate in the blood. It contains various Borrelia DNA. So this pearl may have DNA which is different from the pearl DNA of this end of the string of pearls. These images from Dr. Hoisting and Spearhead and Rosen in the year 2001. When the string of pearls breaks off, then we don't have the tail structure to look at anymore. We have just a string of pearls circulating in the blood, now free in the circulating blood or in the tissue. So it's broken off, and it can break further. It can cut off here and cut off here, so we can have an individual pearl circulating in the blood with DNA, infectious, Borrelia, DNA in the blood, in a pearl. So here we have more breakage. We have more free pearls. Here's a free pearl. It's about to form. We've had a segment that has shortened from 1, 2, 3, 4, 5 to 1, 2, 3, 4 in length, and it can break further. Shorter and shorter strings of pearls in the blood can be seen by fresh blood direct examination using the techniques of Dr. Laane in his laboratory in Oslo, Norway. This is an incredible step forward, an incredible man, incredible diagnostic tool. Now, looking at various types of pearls, we can see they come in different colors. And these different color tools remind us that the Borrelia DNA varies from spirochete to spirochete, and therefore the DNA that is contained in the pink is different from the DNA that is contained in the brown or the yellow. So we should expect DNA variation in string of pearls containing units of DNA from the Borrelia as they circulate through the blood. Size comparisons. The pearls in the tail of the Borrelia are larger than blebs, and I'm going to show you that. Spiral Borrelia, which are not associated with cysts, can rarely form pearls too, but more often we see them coming from cystic forms. Here's Dr. Grossen, Dr. Oystein Grossen here in his lab, working in his laboratory diligently and doing all of the work. that is so important to our understanding of cystic Borrelia, and now string of pearls. This is Dr. Croson's image of string of pearls from his laboratory, and you can see it does look like a jewelry item. These are Borrelia, and they appear, as he said originally, they resemble streptococci. Remember streptococci in the strep throat? There's the chains of bacteria that are connected, and they have a form like string of pearls. They are smaller than the string of pearls individually in size, but they have, at low power, the look of a string of pearls. So this is a Borrelia string of pearls structure from Dr. Croson. Borrelia string of pearls. Now, this is a complicated electron micrograph, and it shows cystic forms here, here. It shows spiral forms here, 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 here. It shows bleb forms, which are the very small, submicroscopic, invisible forms that come off the outer surface membrane of Borrelia. And here we have three pearls that are detached from one another that are in the same environment as the rest of the forms of Borrelia. So the diameter of the pearl is bigger than the diameter of the bleb, but they both contain DNA of the Borrelia. They're both infectious. Pearls are larger than blebs, and they may detach from the string, and they may be single units. So you may have to identify single pearls instead of strings of pearls when you're looking at blood smears under the right conditions. Remember, the DNA is different for some strands, and some of these colors then highlight the differences in DNA that you may see among different origins of the string of pearls in different Borrelia cysts and different Borrelia Spirochetes. Remember the breakage. Single ones can exist, chains can exist. For comparison, the diverse DNA content will be now presented using the acridine orange stain, which I developed in 1989 to show how the spirochete, which is the form that we all recognize, segments and forms what I call a striated or striped pattern, like a barber pole, which has DNA in green or yellow, RNA in red. So DNA, RNA, DNA, RNA, DNA, RNA red, DNA, RNA red, DNA, RNA, DNA, and then a cystic form with both DNA and RNA. Using acridine orange and the appropriate differentiation, we can show that there are zones within the spirochete where the DNA is segmented. And if you cut the DNA here, then all of the DNA will be a particle unto itself, and it may not be exactly equal to the DNA of the particle which separates here, or the particle which separates here. So it offers a chance for different DNAs to get into the blood and into the tissue and cause tissue injury. Now, different DNAs produce different proteins. Different proteins need different antibodies to be recognized. Therefore, how good is your blood test if your blood test is designed to pick up the DNA in this, but not very good for picking up the proteins from the DNA in this or the DNA in this? Blood test is not going to be able to compete. There are too many DNAs producing too many proteins. There is flux. We talked about flux of DNA in the last lecture. Differentiation step is to stain the spirochete with acridine orange at a neutral pH, and then differentiate it by dipping these slides after acridine stain in calcium chloride 10% solution. And that causes these colors to come out. If you do the acridine orange without the calcium chloride differentiation, the spirochetes will look one color. They'll look yellow or green, but you won't see the segmentation of DNA and the RNA at all. You need calcium chloride differentiation to accomplish this. And this is one of my prides, is the fact that I taught Dr. Laane this technique when we first started corresponding, and he's used it very successfully. He was not aware of the technique, and now he uses it all the time, regularly, to look at Borrelia spirochetes. So I was able to share something with Dr. Laane He shared a lot more with me than I shared with him. He is the senior statesman, I'm just the apprentice. So we have zonation of DNA, and it continues into the cystic forms. You can see DNA, RNA, red is RNA, yellow or green is DNA, and then the spiral forms coming off of the cyst. So Dr. Laane's special technique to identify these special structures, the cystic structures, the string of pearl structures in the blood, is now going to be revealed to you. How does he obtain the beautiful images of spiral forms? How does he obtain the beautiful images of cystic borrelia from the blood? How does he obtain the heretofore unheard of images of the string of pearls in Borrelia human blood? How does the man do it? He shared a secret with me. He dips the slides in 45% acetic acid, and the high concentration of acetic acid dissolves away blood proteins and often dissolves away all of the red blood cells. Now, you may do a shorter treatment with the 45% acetic acid and allow some of the red blood cells to persist and give you a structure like we started out with, which is a red blood cell with Borrelia coiled up inside, or you can let the slide sit in the acetic acid for a longer period of time, and then all the red blood cells will be destroyed. Acetic acid is a well-known component of some blood smear fixators, such as carnitine solution, and it is there to reduce the staining or to eliminate the staining of red blood cells. So carnitine solution, acetic acid, removal of red blood cells. Acetic acid is also used in the gynecologist's office to examine the cervix and see if there are what we call white lesions in the cervix. You paint the cervix, which is pink, with acetic acid, not 45%. You use a very dilute acetic acid. If you use 45%, you'll burn the patient. But if you use a lesser concentration, you'll allow the white areas to be visualized from the pink tissue. The pink tissue is healthy, the white areas potentially is a problem. And that's where the gynecologist decides where to take biopsies from the cervix at the time of office examination, using acetic acid to paint the cervix, making some of the tissue look white and some of the other tissues stay pink. Pink is healthy, white is potentially disease. So a recent survey now has been done in year 2010 by Dr. Straubinger at the university in Germany, and he has validated the reality of cystic borrelia. You may ask some senior statesmen in the world of Borrelia science if they believe in cystic forms, round forms. They may say, I'm not sure, I don't know, I've not spent time thinking about it, I don't spend time doing special preparations to see them, and I'm more interested in the spiral form. Dr. Straubinger, past professor of veterinary medicine at Cornell, now senior professor in Germany at his university, he wanted to validate the reality of cystic borrelia, so he did a study. And here's the study. And it's called Metamorphosis. We've heard that before, the metamorphosis of the butterfly, the pupa, and the caterpillar. Borrelia burgdorferi organisms, RNA, lipid, protein composition in context of spirochete shape. And here is a, what is it? Rounded up cystic form with a tail on the cover of the journal Basic Microbiology. So in the year 2010, Dr. Straubinger has validated, that means he put a stamp of approval, validated the reality of cystic forms of Borrelia. And this is what he found. Spiral form undergoes a change where it starts to round up, and then it continues the rounding up process and forms what we all know is a cystic form with the tails. Here are the tails. And at the end of the tails here we have little blebs, and here we may have the beginning of string of pearls. So, encystment, or the formation of cysts, transforms the spiral Borrelia into a rounded cystic form, a round body. It commences in the center of the spiral. Here it is in the center. Continues to round up, and the spirochete coils up on itself and it pulls itself in, and there's the last to enter the cyst are the tail structures, and here they are. And this is an example of encystment, cyst formation. Here's a higher power showing the blebs or the liposomes at the arrow. We know what the blebs are. We discussed those in an earlier lecture. They're released in large number from Cystic Borrelia too, in addition to String of Pearls. And they come from the protruding tail region. So, for review, Cystic Borrelia, first seen in Alzheimer brain, that's me, 1986, extensively studied by Dr. Oystein Brosen, Sperhanning Brosen, and now Dr. Straubinger in Germany. All of them verify that cystic forms of Borrelia are real, they're complex, they have many appearances, they have their own biology, they have their own rules, they have their own lifestyle, and we're going to learn some more about cystic Borrelia now. Cystic Borrelia in Alzheimer brains invade nerve cells. And I presented this work at the Alzheimer's Disease Association International Meeting in 2006 in Madrid, Spain, and published it in Alzheimer's Disease and Dementia, year 2006. Cystic Borrelia invading nerve cells in Alzheimer brain tissue. The following panels now I'm going to use to demonstrate invasion by Borrelia, cystic forms of neurons in Alzheimer's disease. This study is unpublished, but this is a study which I completed in the year 2006. We start by looking at the healthy neuron. Here's the healthy brain, healthy neuron. This is the nucleus. Here's the nucleus of the cell. This, beyond the darker red, the kind of black red, is the cytoplasm of the cell. This is acridine orange without differentiation with calcium chloride. These are Borrelia cysts that have invaded neurons in human with Alzheimer's disease. The Borrelia cyst is in green. And here you can see it's moving its way into the cytoplasm. It hasn't gotten to the nucleus yet. Here it has gone further into the cell and it's disturbed the architecture of the cell, so the nucleus is hard to identify from the cytoplasm. And here it's more or less taken over the whole thing and pushed a dying nerve cell off to the edge. These are invading cystic Borrelia, they stain green, and they're staining nerves in Alzheimer's disease. The cyst can invade nerves. The nerve cells that are invaded wind up being destroyed. Here's the cyst, here's the destroyed remnant corpus, carcass, whatever you want to call it, of what used to be a living, vibrant nerve cell. Spiral Borrelia also invade inside the nerve cell cytoplasm. So we're talking about now spiral forms, not cystic forms, but spiral forms. And this work comes from the Centers for Disease Control, published by Dr. Livingood and Gilmore. And I'll show you some examples of spiral forms. Now, these aren't cystic forms. These are not rounded up. These are spirals. But they're invading neurons, human neurons, and they're invading the cytoplasm. So the spiral form can invade just as the cystic form can invade. And here's the paper, Invasion of Human Neuronal and Glial Cells by Infectious Strain of Borrelia burgdorferi, year 2006, Microbes and Infection. This is a human neuron, nerve cell, human cortical neuron, HCN. The black is the nucleus. The pale green is the cytoplasm. And then the white arrows point to green Borrelia. The box tells us that the arrows pointing to the green Borrelia are Borrelia inside the neuron. They've gotten past the cytoplasm into the inner workings of the cell. They're inside the cell now, maybe inside the nucleus. And then we have a yellow structure, which is Borrelia Spirochetes outside of the neuron. That's this one. It's stuck to the edge of the cytoplasm, so it's cytoplasmic membrane, but because it stains yellow, it has not yet penetrated the cell, either the nucleus or the cytoplasm. This is a classical study of Borrelia invasion of human neurons in the test tube from the Centers for Disease Control, Fort Collins, their leading research laboratory for the United States, Fort Collins CDC laboratory, Drs. Livingood and Dr. Gilmore. Now I'm going to present a case study of nerve invaded by Borrelia, and we'll look at Alzheimer neurons again, because that's my interest, with Borrelia invasion. And this is a nerve cell, and it has a snake-like green thing in the cytoplasm. The cytoplasm here stains orange because acridine orange stains the cytoplasm orange, and the nucleus is yellow. And the Borrelia here, broken up, and here, snake-like and not broken up, are inside cytoplasm of the human nerve cell. This is in the hippocampus. Alzheimer's disease begins in the hippocampus, so if you're looking for where the action starts, look in the hippocampus of Alzheimer's patients and Alzheimer's autopsy brain. These are from hippocampus preparations. These are hippocampal neurons, and they're invaded by the spiral form, not the cystic form, the spiral form of Borrelia. They're in the cytoplasm, just like the Livingood and Gilmore spirochetes are in the cytoplasm. Cystic Borrelia play a role in other invasions of brain, and I'm going to pick on pregnancy because that's one of my second favorite areas of research. Pregnancy and Lyme borreliosis do not always lead to harm to the fetus. So if you're pregnant, you have Lyme, they diagnose it, they treat it. Doesn't mean that your fetus is going to be harmed or that you're going to lose a baby or the baby's going to die. Pregnancy and Lyme borreliosis properly diagnosed and treated can result in a good outcome. And more often than not, it does. However, now, whenever anybody says however, it's telling you that what they just spoke about in the last sentence, they're going to do a 180-degree change in navigation. However means we're going to go in the opposite direction. And that is that blood testing as a tool to discover risk profiles for pregnant women with Borrelia infection often gives no evidence of blood antibodies. All right? No evidence of blood antibodies means you're in the clear? No. Wrong. No evidence of blood antibodies means no evidence of blood antibodies, period. Even when the autopsy of the fetus tells us that the infection has reached the baby and did harm. Autopsy means the patient died. Autopsy tissue tells us that the Borrelia is in the tissue of the dead baby. And I'm going to give you 3 cases to explain the worst case scenario. Now, pregnant women, properly diagnosed and properly treated, do well. Most of the time they do very, very well. Sometimes they're not diagnosed and not treated, and then some of those don't do well, and the babies wind up on my autopsy table. Case number 1, from 1978. Gestational, that means in pregnancy, Lyme borreliosis, implications for fetus. That's my article. Case number 5 out of 14, birth year 1978, baby born alive, died 4 hours post-delivery. Multiple other congenital malformations in addition to hydrocephalus. Hydrocephalus is a very large, swollen brain that's filled with cerebrospinal fluid. It's called water on the brain. These people are incapacitated. And in addition, neural tube defects. As I look at this slide, I see I spelled hydrocephalus wrong. It's Cephalus. So I apologize for the spelling. But hydrocephalus and neural tube defects. Baby born alive, died 4 hours after birth. I did the autopsy. Hydrocephalus, spelled properly here. Live birth, other malformations, died 4 hours later. Case number 1, McDonald, Southampton. The list of malformations includes clubfoot, hydrocephalus, omphalocele, spina bifida, myelomeningocele, and respiratory problems. He had respiratory distress and died in a newborn nursery 4 hours later. This is a spirochete using a monoclonal antibody, 9724. 9724 tells us that the spirochete, which takes the antibody stain, has proteins that the monoclonal antibody recognizes as Borrelia proteins, confirms Borrelia as the type of spirochete. This came from the brain. Dead baby, hydrocephalus. McDonald, 1978. Case number 2, hydrocephalus in a developing fetus caused by Borrelia infection in the womb. Second case. Where did it come from? McDonald, 1989, Southampton. Here's a hydrocephalus. It's a bag of water, which is cerebrospinal fluid, squeezing the brain so that it's paper thin. Here is the brain, the pink stuff at the edge, a very thin brain. And this patient, this fetus, didn't make it. It was miscarried. And the hydrocephalus was apparent. Here it is. And the fluid inside the brain gradually destroyed neural tissue. So you see in this case, which I'll show you just in the next slide, this is case number 2. We're still with case number 2. Hydrocephalus, Cystic Borrelia, participated in the brain injury. And here they are. Cystic Borrelia number 1, number 2, and number 3, all reactive with our friend 9724 monoclonal. Now, I'll tell you a secret. I took this slide many, many years ago, and I always looked at the slide and always marveled at how beautiful this long spirochete is. And for many, many years, I ignored this and this and this. There's 3 of these. There's only 1 of these. But I liked this. I thought it was pretty. And I ignored this and this and this for many years until it became clear to me that 9724 causes things to glow under the fluorescent microscope only if they contain Borrelia proteins. So these are Borrelia cysts from the brain preparation, 9724, from the brain of the baby who had the hydrocephalus. And they coexist, that means they exist at the same time, with elongated Borrelia from the fetal autopsy brain. Here's elongated Borrelia. And here's the rest of the story. This is the baby. This is the brain, hydrocephalus, the 3 cystic forms, the elongated spirochete, and then this is the fetal heart. Now here's a question for you. Can you count the actual number of Borrelia spirochetes which damage the heart of the baby? This is one section. This is 3 microns thick. Can you imagine how many more spirochetes were in other parts of the heart? We only took a sample of one section with one slide and stained it and looked at it under the microscope. There are thousands of other slides that could be prepared from the heart. All of them probably laden with, burdened with, overwhelmed by these Borrelia spirochetes, which stained with not the 9724, but the 5332 monoclonal antibody, absolutely specific for Borrelia burgdorferi. This baby died of intrauterine Lyme disease. This is the rest of the story, as Paul Harvey would say. The rest of the autopsy. It shows the heart also involved with the infection that destroyed the brain by producing hydrocephalus. Case number 3. This occurred 16 years after case number 1 and 2. Those cases I diagnosed and wrote up. This case came from Turkey, and the year was 2005. It was an infant with hydrocephalus detected by imaging studies in utero. The baby was born alive. A shunt was placed to treat the hydrocephalus. The baby is still alive. And postpartum, that means after delivery, the mother's blood was tested and was positive for Borrelia antibodies. So they wrote it up as gestational Lyme disease. That's my title, gestational Lyme borreliosis, as a rare cause of congenital hydrocephalus, disappearing 16 years after I wrote up my autopsy study from Southampton in 1989 in the Turkish-German Gynecological Association. This is case number 3, hydrocephalus and fetal Borrelia infection, fetal borreliosis from Turkey. And here, this is all the cerebrospinal fluid, vastly large in size, creating these rounded areas and stretching the brain to its limit. Triventricular hydrocephalus in the living unborn child. And the arrows detect the hydrocephalus fluid here, here, and here. So we have triventricular accumulation of fluid causing brain injury. But the patient was alive. This is the rest of the baby. This is the spinal cord. This is the feet, and this is the head of the baby right here. See how big the head is in comparison to the rest of the body? Another view with another imaging study showing the same thing with lots of fluid, a very big head. This is the uterus, and this is intrauterine diagnosis of hydrocephalus. They did a cesarean section. If you tried to get this big head through a normal-sized birth canal, the baby would probably die during an attempt to deliver it vaginally. So they did a cesarean section to deliver the baby safely from the abdominal approach. And our studies, Western blot analysis, high specific antibodies are found in maternal blood, and Borrelia burgdorferi, non-specific antibodies are found in neonatal blood. That means in the baby's blood. The antibodies in the baby were not as specific. They were Borrelia-like. Antibodies in the mother's blood were highly specific. So here's a disconnect. The blood of the baby with a major league infection and a hydrocephalus has non-specific, not so much, Borrelia-type antibodies. The mother has straight ahead, Western blot positive, highly specific antibodies in the mother's blood. What does that tell you? That maybe the Borrelia that got to the fetus had a different DNA from the Borrelia that was in the mother's blood to which the antibodies were produced. Again, DNA in flux. Two different DNAs, two different antibody patterns. All right? Not so specific here. Non-specific. Here, highly specific. Maternal blood. Baby, not so much. But the baby had the major league disease. So we have intrauterine infection of the fetus with complications of fetal hydrocephalus. 3 cases now published in the world medical literature. They're summarized here. Case 1, McDonald. Case 2, McDonald. Case 3, Turkey. All in common, Borrelia got to the baby while the baby was in the uterus and developing. Borrelia infected the brain. Borrelia created swelling and then blockage of cerebrospinal fluid, hydrocephalus, and death in 2 babies, and 1 baby survived because of very good medical care and prompt cesarean section. 3 cases, Borrelia infection in the uterus, hydrocephalus, a rare situation for any fetus. 3 cases in the world's literature. 2 of them are mine. Just a fluke? Well, what is a fluke? A fluke, according to the Oxford Dictionary, is that, in a sense, a successful stroke, maybe a golf swing, made by accident or chance. First coined word in the year 1857. So we talk about things being flukes. It means that, not much there to support it. Well, 3 cases overturn the idea that we're dealing with a fluke. We're not dealing with a fluke. We're dealing with 3 human cases. This is reality. And in one of the cases, Cystic Borrelia and Spiral Borrelia formed a prominent role in harming the baby's brain. So we're going to have a hypothesis now, and that is, what are the round things that are seen in neurodegenerative diseases in the human? Are they cystic borrelia in dead and dying neurons? Well, I wrote a paper about this in 2006, and the paper had in its title, in short, Cystic Borrelia hiding in plain sight. So I thought, after looking at many slides and reading many books on neurodegenerative conditions in the human, that round things were turning up inside of diseased neurons very regularly in a number of conditions. Those conditions were amyotrophic lateral sclerosis, Lou Gehrig's disease, diffuse Lewy body cortical dementia, which killed my grandfather, Parkinson's disease, that's the movement disorder that Michael J. Fox has and other people, Neurodegenerative diseases, Parkinson's-like, but not exactly like it, Pick's disease, a degeneration of the frontal lobes, the brain, and the muscles, muscular system, and frontotemporal dementia, which is second only to Alzheimer's disease in terms of its impact on dementia care in the United States today. All of these neuropathology disorders, all of them, I think, can be put into a round body neuropathology container. This is a proposal. It's not proven. But take a look, see what you think. All of the neurodegenerative diseases here have round bodies inside the diseased nerve. And just to remember, this is the work from Alzheimer's showing the cyst invading the healthy nerve with the cyst in green and the nerve in red. So cysts invading healthy nerve. Here is a diseased nerve with a round thing. Here is a diseased nerve with a round thing. These are black staining round things inside diseased nerves. A pink staining round thing inside Lou Gehrig's disease neuron. Round things, brown and black, inside diseased nerve of FrontoTemporal Dementia. And round things here and here, invading the nerve from the outside in Parkinson's disease. Are they Borrelia cysts? I wonder. In at least one case, I have studied the changes of cortical Lewy body disease, and I found that the round structure of cortical Lewy body disease stains positive with a DNA probe for the DNA of Borrelia. That's one case. But that's one case which increases my interest in studying other cases of neurodegenerative diseases in this category: Parkinson's, frontotemporal dementia, ALS, Pick's disease, CorticoBasal Degeneration, to see if their round bodies also light up like these do. And that would prove that they're cysts. And that would prove that these degenerative diseases are not degenerative for unknown cause, but they're related, if I'm right, to infections that have penetrated the nerve, invaded the nerve. And we know Borrelia can invade nerves. We've proven that with Livingood and Gilmore. And once inside the nerve, kills the nerve from the inside out. Inside out. Right. So we don't know for sure whether all of these could be root causes for round body diseases in the neuropathological literature and degenerative diseases, but it's tantalizing, and I'm continuing my studies to take a look at these possibilities. So summary: round body Borrelia, cystic, they're reversible. They can resume the spiral shape. The spiral forms the round body when adverse situations occur and the spiral isn't happy, so it becomes rounded up and protected in this cystic form, round body cystic form. Bad behaviors of cystic Borrelia. They persist in living cystic Borrelia in hostile conditions, so they become cystic when these conditions are hostile. There's multiplication of cysts. The cysts can divide like ordinary cells. You can have one cyst that divides into two parts, just like a normal healthy cell, and you have two cysts then. And I think cystic Borrelia can invade brain and nerve cells, and because they can invade and push from the outside into the nerve cell or into the brain, it implies to me that there might be some motility or ability to move associated with cystic structures. This idea also needs further research. It hasn't been proven. We now think of cysts as being like rubber balls floating in a swimming pool and not able to move on their own like an outboard motor would move structure across the swimming pool. But perhaps the cysts can move. Reproductive activity. Here we have spiral Borrelia dividing in two. Cystic Borrelia can divide in two. Spiral Borrelia can fragment and form granules, up to 40 infected granules from a single spiral form, each of them separating here along these arrows to form a granular form. Each of these granules infectious. Cystic Borrelia can also form granular cystic Borrelia forms. Here, granule forms inside a cystic Borrelia. These granules are like these, except these granules are surrounded by a wrapper, the host of Dr. Laane, the cyst wall of Borrelia cystiforms, round bodies. Now, cyst forms can show cell division, and Dr. Brosen proved this. So another kudo for Dr. Brosen, Spearhenning, and Dr. Oystein Brosen, the two cousins. Here we have a cyst, we have a straight line, we have a D. That means that on this side of the line and on this side of the line, the cyst is about to divide into two parts. And it's dividing. D means dividing. Here we have more evidence of dividing inside a cyst. Only the spiral forms are dividing here. So here's the nucleus of the cell. It's not dividing into two, but the spiral forms that are with the cystic nucleus are dividing. So we're making more spiral forms. Here are flagellae. Flagellae are what make the Borrelia move. Flagellae are the little ropes that are connected to little motors that make the Borrelia move in body and fluids. They are the agents of motility. They're like the outboard motor for Borrelia. Now, normally, the Borrelia flagellae are inside the region between the cylinder or the spiral form and the outer surface membrane, which is here. But in the cyst, they are released. They're still flagellae. They're still alive and they're still viable. And if they're alive, perhaps they can do their one and one only function, that is to move the spirochete. So why not have flagellae move the cyst too, but from the inside? Flagellae inside a Borrelia cyst. And another picture of Borrelia cyst. Here are the cylindrical forms. Here are the flagellae forms, the little motors, the outboard motors. Here are more dense forms within a large cyst. And these little rope-like things are the flagellae. So you can see that cysts, in many cases, are endowed with a large number of flagellae. And flagellae have one function on Earth and one function only, and that is to move, to twitch, to do motility, to be an outboard motor. So they can maybe can move the Borrelia by twitching inside the cystic form as well as twitching inside the cylindrical form. And maybe there is motion associated with Borrelia cysts, just like there is motion associated with spiral forms. We don't know, it needs to be proven. So cell invasion, spiral Borrelia, yes, they move. Spiral Borrelia wind up inside nerve cells. They wiggle their way in. Cystic Borrelia, motility, maybe not. But maybe. This one's pushing itself into a nerve, this one's pushing itself into a nerve, this one's pushing, and that one's pushing. So they're able to invade human cells. So perhaps there is a motility possibility for the cystic forms that are invading these nerve cells. In order to invade, I think a cystic form must have the ability to move to get the invasion job done. This will not move further into the cell unless there is some sort of an outboard motor or a propeller or a flagellae that's moving it inside the cell that it's attacking. This one hasn't gone very far, but it's headed this way, into the cell. Blood testing may fail to detect cystic Borrelia proteins. This is a shocker. It has been found that one of the major proteins of the spiral form is called outer surface protein A. That's one of the major things that's looked for in blood testing for antibodies in Lyme disease antibody testing labs. This protein A. Cystic Borrelia have no protein A on their surface. That means if there is no protein A on the surface, there is no opportunity for an antibody to see protein A and to make an antibody to protein A, which means that the membrane is probably turned inside out, and we can see the protein A is now on the inside of the cyst. And spiral Borrelia, becoming cystic, internalizes, turns the membrane inside out, and puts the OSP-A inside the cyst wall, where it's not exposed to the immune system and antibodies can't be made to it. It probably takes the other major proteins with it too. So if you have a cystic infection with very few spiral forms, you may have a negative antibody result. What a shocker, right? It's taken its proteins and it's removed them from areas that the antibody system can see and recognize, and it has what we call sequestered them. It has hidden them. It has concealed them. It has turned them inside out. And when they're inside, the immune system can't make antibodies to them. So here we have, again, a look at the shortcomings of the antibodies in blood. The efficiency of blood studies in Borrelia diagnosis, not so good. Many cases never produce an antibody, which can be measured by either ELISA or Western blot. Many more cases produce low-level antibodies, and it's not high enough by cutoff, by laboratory methods, and precise analysis, precise regulation of the testing, not high enough to confirm Borrelia infection. So you have low-level antibodies, they are meaningless. Or are they? And then in many, many more cases, antibody production is interrupted. If you get antibiotics early on, the antibiotics may kill off a lot of the bacteria, and then there's not anything for the antibody-producing cells to make antibodies to. Or if you are immune depressed, you have cancer, or you have steroid therapy, you will have interruption of antibody production. Or if you have lots of co-infections like babesiosis, or adenomyosis, or anaplasmosis, all of the co-infections of Borrelia are immune suppressive, and then they interrupt the production of antibodies so that the antibody studies will not give a high enough level to call the blood test positive. In chronic Lyme borreliosis, the CDC has even had the declaration, I'll call it, that even positive antibody tests, when you're dealing with a person with chronic Lyme borreliosis, somebody who's had infection and symptoms ongoing for more than 6 months, that's chronic problem. Even positive antibody tests are no longer counted in chronic Lyme patients by the CDC. You can probably get a better answer from them than I can about that, but for chronic Lyme, the rules are different, and even a positive antibody test doesn't count anymore. So the intrinsic limitations of the use of one Borrelia strain, as what we've talked about before, the B31 strain, used to manufacture all USA test kits, is a final limiting factor in the efficiency of antibody studies in Borrelia disease diagnosis. And so we are entering now the closing part, and we have to acknowledge copyrights. Image data here is from sources duplicated with publishers' permissions from Dr. Grossen or Dr. Laane, or his special permission given to me verbally to use some of his photographs in this presentation. Some of my publications are protected by copyright. When I publish them, I have to give the copyright to the publisher. And so duplication of these materials is not allowed under copyright law. The presentation is just for educational use. It's free. It's not for resale. And all the references to this material are available on our website, which is www.molecularalzheimer.org or www.alzheimerborreliosis.net. I have two of them now, and I may have a third. So you'll have the references there if you want to go and look at them afterwards. This concludes the lecture, and I thank you very much for your kind attention. I hope that you have learned something, and I hope that you will continue to be curious and ask good questions.